So in this video, we're going to talk about the digestion of proteins, which is pretty similar to the digestion of carbohydrates, breaking it into smaller and smaller pieces and then finishing off the digestion with brush border enzymes and then absorbing into the cells and then into the capillaries. Um, so it's going to be a lot of similarities, different enzymes and different beginning places. So proteins broken down to large polypeptides, so those long chains of amino acids, and slowly we're just getting to smaller chains of amino acids. So small polypeptides and small peptides, so even smaller, to eventually remember the building blocks of proteins is chains of amino acids. So there's 20 amino acids that we make a combination of in chains to make a polypeptide and eventually a protein. Sometimes we can have dipeptides and tripeptides, so 2 amino acids um, and 3 amino acids. So sometimes it can be that small, but most often it's going to be those amino acid monomers, those building blocks. Now, unlike digestion of carbohydrates that begins with salivary amylase in the mouth, digestion of proteins begin in the stomach. So not only is the hydrochloric acid, so the pH of 1.5 to 2.5-ish, is going to denature the proteins. So remember, they have that kind of final shape. So we had the primary, which is the amino acids, secondary, which is the beta pleated sheets and the alpha helix, tertiary, where we start kind of folding in a 3D section, and quaternary, where there's multiple polypeptides chains interacting in the protein. So first off, the pH will make it so that the enzyme can get to it. So it's going to unfold, make it so it goes from its globular or kind of shape, the folded shape, to more a string so that enzymes can start breaking it down a little easier. It's easier for them to get in and do their job this way than it is when it's this way. So digestion begins with that, and then also in the stomach. Remember, pepsinogen is secreted by chief cells, but they need parietal cells as well. So pepsinogen is produced by chief cells and released, but without the parietal cells producing hydrochloric acid, it's never converted to pepsin, which is the active form. So remember, pepsinogen inactive, pepsin active, and it's activated by the low pH of the hydrochloric acid that's produced by parietal cells. Once the pepsin from the stomach is kind of breaking down proteins and it goes into the duodenum, then we have a higher pH and pepsin is inactivated again, so it's no longer working. In the high pH of the duodenum, remember we add, the duodenum can't handle this really low pH, so we add bicarbonate from the pancreas mainly, and we make the pH higher, which is going to inactivate pepsin. So looking, and now we've gone from the stomach into the small intestines, we add pancreatic proteases. So these ases are going to break down proteins. So we're going to break them into smaller peptides. Carboxypeptidase is a pancreatic protease, and it's going to kind of chop one amino acid out on a time from the end of the chain. So it's just going to chop the ends kind of slowly, just chop, 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 chop. And then same that we saw with oligosaccharides and disaccharides for the brush borders, we're going to have the brush border enzymes in the proteins break down oligopeptides and dipeptides, so kind of break them down into their building blocks of the monomer of amino acids. Just like we saw with the monosaccharides, the amino acids are also co-transported across the membrane. So again, the apical membrane is the brush border with all the microvilli. So you have this, and then basolateral side is this side with a capillary here. So we still have the same way that we discussed with carbohydrates, the secondary active transport. We have a sodium potassium pump that's pumping sodium. Sometimes we can have hydrogen as well. So pumping something out actively, this example, sodium. So it's pumping sodium out, and then sodium travels down its concentration gradient again and brings an amino acid with it. Then we have the same thing at the basolateral side. There's channels for amino acids. And so then they can travel from high concentration to lower concentration. So high to low. And they'll be able to travel into the capillaries to be absorbed. So facilitated diffusion, remember, is still simple diffusion. You travel from high to low, but it has a channel or a carrier that helps it go through the plasma membrane. So same, again, we have peptides here that are broken down by the brush border enzymes. So these are small peptides that are broken down to amino acids, broken down by to this level by the pancreatic proteases. And then we have sodium that co-transports the amino acids into the cell. So brush border enzymes break it into amino acids. We have the sodium potassium pump over here that's going to make the sodium concentration gradient. And then we have a facilitated carrier down here that's going to allow diffusion from high concentration over here to low concentration here. Still looking at it, so pepsin is going to start. Um, so pepsinogen released by chief cells, activated by the hydrochloric acid produced by the parietal cells, and starts breaking down proteins into large polypeptides. The pancreatic enzymes, you have trypsin, chymotrypsin, carboxypeptidase, that's going to break them down even further. And then the brush border enzymes are the last ones.